Hello, my name is Dr Nicholas Ashton. I'm from the University of Göteborg in Sweden. I'm a neuroscientist trained at King's College London, and I got my PhD also from King's College London. My interest in Alzheimer's disease has stemmed from fantastic mentorship. I had a PhD supervisor, Professor Simon Lovestone at King's College London. He set this challenge to me to try and diagnose a brain disorder without touching the brain or even looking at the brain. We needed new tools to understand who had Alzheimer's disease and who did not, without going into complicated and expensive brain imaging. So the idea we started working on about 15 years ago with this idea that we could take a simple blood test and try to understand who had Alzheimer's disease and who didn't. This started off with a lot of complex, explorative blood proteomics. But now, a decade later, we now have really robust blood tests which are starting to be used by doctors to understand when someone has symptoms, is this caused by Alzheimer's disease or is this caused by something else? So after completing my PhD and doing a postdoc in King's College in the United Kingdom, I was recruited to Pr Zetterberg and Kai Blenau's lab in Göteborg in Sweden, a world-famous lab for fluid biomarkers. At that point in time, we were really still looking at cerebral spinal fluid biomarkers for Alzheimer's disease. The challenge when I arrived there with my colleagues was to try and translate some of these CSF tests to blood tests. So we started that journey in 2018, and as I say, from today forward, we have really robust tests which are being used worldwide now in clinical trials, but also by doctors in clinics as well. We spent years trying to come up with protein signatures in blood by using mass spectrometry and unbiased proteomics. But what we've been able to do because of the advance of technology is essentially detect the same proteins that we look at in cerebral spinal fluid, but now in blood. But these are much more dilute, so femtomolar levels, a billionth of a gram in concentration in the blood. So we use techniques called single molecular detection, so this detecting single molecules of phosphorylated tau and other proteins called neurofilament light, which help us understand whether someone has Alzheimer's disease, and neurofilament light helps us to understand about neurodegeneration. So it's been a real advancement in our technology. Also, it's been an advancement and a revolution in the way that we diagnose Alzheimer's disease. Previously, it was purely based on clinical symptoms, and now it's more guided towards a biological characterization. So we have great cohorts and research studies which have amyloid PET and CSF, and that gives us a really fantastic way to see if our blood tests are working well or not. Next step in my research is I've now started my own group in Arizona, United States at Banner Health, and what we are trying to do there is implement these blood tests that we have on wide scale. So this would be in clinics, also to help clinical trials. We're also seeing how these blood tests would help in primary and secondary prevention, so individuals that do not have cognitive symptoms but are at risk to developing Alzheimer's disease. Alongside this is that Alzheimer's disease or Alzheimer's disease pathology is not the only pathology that is causing dementia. We have many other types of proteinopathies, and currently, as we stand today, we don't have any blood tests or cerebral spinal fluid tests for TDP-43 or alpha-synuclein. These are very common proteinopathies that go alongside Alzheimer's disease and cause other types of dementias like Parkinson's disease and frontal temporal dementia. So my research in my new group over in the United States is to try and develop similar types of blood tests, but for these other proteinopathies. So when someone comes into a clinic, you can say, not only do you have Alzheimer's disease, but you're also at greater risk because you have TDP-43 pathology, or this isn't Alzheimer's disease, this is a different type of dementia. And that gives us a much better personalised care and personalised management for that patient. The idea at the moment is that we are waiting for someone to get mild cognitive impairment and a lot of amyloid pathology in the brain before we treat. Now, there is a hypothesis is that the earlier that we treat, the better the prognosis of the patient is going to be. Our aim is that we start clearing amyloid plaques before the symptoms arrive, and maybe you'd be able to stave off the symptoms even coming, or at least at a later date. So this is where a blood test becomes extremely important, because you don't have cognitive symptoms. The individual or the person who is at risk doesn't know or experience that they have cognitive complaints. but a blood test will be able to say that they have pathology in the brain, and there we can start some treatment. So there are ongoing trials right now testing whether these drugs that have been approved by the FDA and the EMA would work in people at an earlier stage of the disease. And if that proves to be effective, then the blood test becomes essential in understanding who gets that drug and who doesn't. The question was, do we have biomarkers that can help us understand progression? I would say that phosphorylated tau, which is the biomarker that I've been talking about, is our best marker of progression, but it's not a very adequate one of doing so. So we are still trying to understand if someone has high phosphorylated tau in the blood, which indicates that they have pathology in the brain, who is likely to progress quicker or not. And that's also a part of the ongoing research, is trying to understand what other elements to the disease cause progression. Is it something like neuroinflammation, which is some hypothesis to say, do we need to treat an underlying neuroinflammation? Does that cause the person or participant to progress quicker than someone else? So this is all ongoing research as well. So the question is, do we need to treat multiple proteinopathies? I think the answer for that, unfortunately, is yes. But I think that if we can treat amyloid pathology at the earliest stage, then maybe you're less likely to get multiple pathologies later on in life. It's very clear in Alzheimer's disease that these other pathologies come later. Tau, TDP-43 alpha-synuclein, they come later on in disease. So being able to stop the catalyst of this, which is amyloid, would maybe prevent these proteinopathies from forming until a later stage. Now, if we're talking about individuals that have symptoms right now, this kind of dual targeted approach is probably where we need to be. There are already ongoing trials that are combining amyloid and tau therapies together to see how they have greater therapeutic effect. And we may need to understand the similar thing with TDP-43 and alpha-synuclein, but it becomes very difficult to come up with these new therapies if we don't have an effective biomarker to diagnose them in living patients. So that's why these blood tests and cerebrospinal fluid tests of TDP-3 and alpha-synuclein become critically important. So I would start off by saying I think that we're in such an amazing point in Alzheimer's disease and dementia research. We have, as I said, blood tests that can help a clinician understand whether Alzheimer's disease is causing symptoms or not. We have the first disease-modifying therapeutics approved worldwide for treating Alzheimer's disease pathology, and while they're maybe not the silver bullet, they are definitely effective and they're getting better and better. So at this particular moment, there's no better time to be in Alzheimer's disease research. I think that what we will start to see is, as I've discussed, is that treating people earlier in the disease is likely to have greater benefit, and we will know the readout from these trials in the next few years. I also think that we're getting better at diagnosing patients. We're getting great research cohorts, so understanding this idea of co-pathologies. Do we need to treat neuroinflammation? Do we need to understand if someone with TDP-43 pathology should get an Alzheimer's drug or not? And these are all answers we are working on. We should have the answer to in the next few years. But right now, as we stand, we can diagnose patients through a blood test, and we have a pathway where they can receive a treatment which has benefit. And I think that's a really exciting place to be. So this prize is a fantastic honour. I see who's won it previous to me and how they've gone on for their independent career, and I hope to follow somewhere similar, starting, as I say, now at Banner Health in Arizona. And it gives it's a terrific recognition to not only my work, but everyone in the team and my colleagues that have been working on this idea of a blood test for over a decade, starting right from the start, where it was seen as very much academic science, right today where clinicians are using it on a daily basis. So this is a tremendous recognition, as I say, not only for me, but for an international effort for blood tests, because they are really going to revolutionise research and drug trials and clinical practice. And as I say, now I will use this as a stepping stone to continue my own independent research, to continue the idea of being able to treat Alzheimer's disease and diagnose Alzheimer's disease and dementia using simpler blood tests.